This is Audiommunity, a podcast about our body's never-ending fight with the outside world.
Hi everyone, this is Audiommunity, recorded on March 21st, 2018. I'm Kevin Bonham, and I'm joined this evening by Kate Franz.
Hello.
And Chadene Tremeglio.
Hello.
And Camilla is off, I think, traveling the world, and Matt is too busy for us. Matt's pulling a Kate tonight. But I still think we're going to have a great discussion about something that is near and dear to Kate's heart, and that's the antiviral receptor, maybe? Sting? Not exactly a receptor.
I mean, it's a receptor for CGAMP, but it's in like its traditional pathway is a signaling adapter. So it induces transcription.
Well, we'll get there.
Yeah, we'll get there.
But before we do, Kate, what are you drinking this evening?
I am drinking TW Pitchers Lager and Grapefruit Radler.
Ooh,
what?
That's interesting.
It tastes like blood orange soda, so it's delicious.
Yum.
It's something that my husband bought because he doesn't like the taste of beer. It mostly tastes like soda, but it's really good.
Okay.
We were watching Top Chef, and one of the challenges earlier in the season was to pair a meal with a Radler, and so it like sparked something in my husband's brain. He was like, "Oh, I have to get some. We have to try this." So it's like a nice, entering into like cookout season sort of beer.
Definitely.
Mm-hmm.
Definitely.
And it's 5% alcohol, and so that's like the tops alcohol I can drink in a beer. So it's like right at the perfect spot for me.
Well, that sounds kind of gross, but kind of good. I don't know.
It's delicious.
It sounds great.
And what are you drinking, Chadene?
I'm the loser with tea again. I'm sleep training my 2-year-old this week, so I'm exhausted. FYI, if I say anything stupid, that's the reason. Unlike last time, well, I was going to say, unlike last time where I pronounced La Jolla as La Jolla, that was just straight up pure unadulterated ignorance. But this time I have an excuse, so.
I cut that out. You didn't have to throw yourself under the bus there. But
there you go.
We'll leave it in.
Oh, well.
That's great. This evening, I am drinking a Mayflower oatmeal stout.
Oh,
I
yum.
love that.
That's a good one.
Which is like just really straightforward but delicious stout. It was supposed to be a blizzard day today, and I stayed home from work, but it still hasn't started snowing here in Boston. It's a little gloomy, but other than that, it's just fine. So hopefully we're not going to get dumped on too badly. But I still thought the stout was appropriate.
Awesome.
And you have no response.
No response.
Great.
I mean, sure.
So with that awkward response to my beer drinking out of the way, maybe we should just launch straight into the paper. So the paper that we're doing this evening is a paper that is sort of related to the work that one of us did. There's this first author, Kate M. Frains, maybe?
Yay! Yeah, it's my paper from grad school.
Woo!
Woohoo! The majority of the work... see, now here's a question. Is this representative of the majority of the work I did in grad school? Or the tiniest sliver of the work I did in grad school?
Well, if my PhD is anything to go by, I would expect that it's the tiniest sliver, and the vast bulk of it was trading drives with Charlotte, or having Western blots that didn't work, or getting scooped.
Yeah, I was going to say, like, getting
having
scooped.
Artifacts.
Like, this paper was, like, trimmed down from, like, 14 full-page figures.
Oh, wow.
Oh, yeah.
That's intense.
Oh, boy. Yeah, we were told by multiple people, you should split this into 2 papers. And I was like, no.
They go out
One
together
paper.
or they don't go out.
Yeah. I can't go through this twice.
No. Nope. It's a really beautiful paper, though. It's really nice paper.
Aw,
Good job.
Thank you.
I think this might actually be the first time that I've read it. No offense, Kate.
No, you think I read your paper? No.
That's fair.
Burn.
Good point.
Actually, side note, once in a student-run data club, I in the back of this is like how much attention I paid while in lab. In the back of my head, I knew that TRAP was important for TLR9. Couldn't figure out why, and so I was like I was like giving someone feedback, and I was like, you need to like write this like this TLR9 pathway properly. Like you need to put TRAP in there. And like this person gave me so much freaking attitude, and like someone like did all this Googling, and they're like, look, like TRAP isn't here, TRAP isn't here. And I was like, oh, okay, cool. I don't know why I thought it was. My bad. And I was like, damn it. I knew there was a reason why I thought it. Someone in my lab is researching this and the paper came out. Okay.
Yeah. Yeah. Well done, Kate.
You're welcome.
Kate is referring to the fact that the paper that I published in grad school basically showed that this protein, TRAP, that was involved in a signaling pathway that people didn't think it was involved in. That was a real inside baseball reference there, Kate, but I'm going to leave it in.
Yeah,
and people can just scratch their heads over it.
Go PubMed, Kevin Bonham, look for something about promiscuous lipid binding, and have at it. You'll see exactly what I meant.
How do we transition from that to the paper?
Well, I mean, like, we can talk about, now that you asked, like, I don't know.
Right, this is what happens.
I was just going to say, how about speaking of things that nobody were certain were involved in certain pathways, I thought this paper was really cool because it took a factor that people already believed to be involved in one specific pathway and expanded its role into other pathways. No?
Yeah, I think that'll work. Yeah, I mean, this is sort of the bread and butter of the Kagan lab that both Kate and I graduated from, is sort of doing sort of deep biochemical dives into signaling pathways associated with the innate immune system, and ideally pulling random obscure things into the light that other people either didn't think to look at or dismissed or sort of ignored for other reasons, and make them awesome.
Nice.
How do you think John Kagan would feel about you characterizing his research strategy as pulling random and obscure things into the light?
I'm not sure that he would endorse it full-throatedly, but on the other hand, that's because John is sort of an expert at spinning things to make them seem really awesome. Not that they're not really awesome.
We're gonna cut this.
Really awesome. It's just that
keep digging that hole.
Rather than going like, oh, everybody else is studying checkpoint inhibitors, I'm going to study checkpoint inhibitors too. John will take something and be like, you think checkpoint inhibitors are cool? Check out this other thing that no one is looking at, and here's why it is better than checkpoint inhibitors. That's how I think of John.
I think like the research that Kevin and I did was very much based on this idea that given like the way the literature is laid out, we can draw a set of conclusions about the way these pathways should function inside of the cell. And we test those and we look for the inconsistencies, and then we take that, iterate, and refine the rules. And we find, and like there's been a history within the lab of finding like really cool regulation motifs or different structures that really couldn't come to light until you put them under the right stress. And so I think that's where like that lab, like John's lab in particular, thrives, is like looking at the expectation and then like finding, like testing it and then finding something like novel or something interesting about that.
I agree with that completely. And that was a much nicer way of putting it than the way that I put it. But so with that in mind, so what's the sort of paradigm or scope of what you were looking at, and what were the inconsistencies or gaps that you noticed?
So just like to focus us on the scope of the question. So my research focused on how the control of viruses is initiated by the pattern recognition receptors and their pathways. So like specifically looking at the CGAS sting pathway, which detects DNA viruses, and the RIG-I MABS pathway that detects RNA viruses. So how do these pathways, like how are they activated and how do they control viral replication? And so basically the dogma of the field is that control is initiated when a PAMP, so that's a pattern or a pathogen-associated molecular pattern, it's been a while, is recognized by a pattern recognition receptor. So there's a piece of the virus that binds or is recognized by a protein in the host, which is called a pattern recognition receptor. That receptor signals downstream. And in the case of viruses, we mostly think of the initiation of interferon as being protective and being required for control. So you can think of this as there are like these two parallel pathways so cGAS and STING, or RIG-I and MABS. cGAS binds to viral DNA, so it recognizes DNA viruses. And RIG-I and the RIG-I-like receptors bind to viral RNAs, so it recognizes RNA viruses.
And just to back up for a sec,
yeah.
This is happening inside the cell as opposed to being a receptor that is facing the outside of the cell. So a lot of pattern recognition receptors, the one that I was most focused on in my PhD, is like most cellular receptors, or a lot of cellular receptors anyway, sort of embedded in the plasma membrane and facing towards the outside of the cell.
Yeah.
So these receptors are different in that they're in the cytosol. And so they're basically detecting viral RNA and DNA when the virus has like entered the cell and uncoded and potentially is actually transcribing genes, right?
Yeah,
its genome is in the cytosol of the cell.
Yeah, so the idea is that you want to position pattern recognition receptors where pathogen replication is occurring. That's like a broad theme in innate immunity. So you can have, and you can, I mean, there are receptors that are extracellular facing that can detect viruses, but you could hypothesize, and there have been reviews written on this idea, that you want to put receptors where the virus, that it's active, it can replicate, it's competent, and then that's a better sensor for, or it's a better way to control your responses to your antiviral responses. So the external ones like that detect bacteria. If a bacteria doesn't necessarily need to be inside of a cell to replicate, so it makes sense to put like these bacterial responses externally. Getting back to like where I was with my story was, so we have these parallel pathways. They detect two different types of viruses, and I just want to highlight that. So DNA virus, we've talked about this before, but when I say DNA virus, I mean a virus that encodes its genome in DNA. And when I refer to an RNA virus, I mean a virus that encodes its genome in RNA. DNA viruses will, of course, make RNA, and those RNAs, under certain circumstances, could be detected by RIG-I. But what we want to focus on is these pattern recognition receptors need the correct PAMP in order to be active. So if you have a DNA virus, it's going to be detected by cGAS and STING. And if you have an RNA virus, it's going to be detected by RIG-I and MABS. And so because RNA viruses don't have DNA, you would not expect cGAS or STING to be necessary for the control, because cGAS and STING will not induce interferon in response to RNA. And so our idea of how control is initiated doesn't occur.
Can you just describe briefly, I know it's not the focus of this paper, but like what cGAS is doing? Because I think that's a cool story. And we might have even done a paper on this years ago, but maybe. I think it might be nice to refresh.
So what is cGAS doing? So cGAS is a cytosolic protein that binds double-stranded DNA. It binds B-form DNA. So it can also bind DNA-RNA hybrids.
Oh,
sorry.
Cool.
But when it binds DNA, there's a rearrangement within the protein that allows it to synthesize cyclic dinucleotides. And cyclic dinucleotides are really interesting. They're very involved in bacterial signaling. But cGAS actually makes these. So cGAS is the first protein that's known to make cyclic dinucleotides in metazoa. So when it was discovered, it was this huge discovery. There was actually like many years of research trying to determine what is the DNA sensor, what activates interferon in response to DNA. And so cGAS was originally described in late 2012 by James Chen at UT Southwestern. And so the cGAMP molecule that cGAS makes, it's in the cytosol. It just thought probably just to diffuse to sting, which is a protein in the ER that binds cGAMP, and then it can activate interferon.
Cool. Yeah, I just think it's a kind of a neat mechanism. There are some other signaling pathways that we often learn about in like biochemistry class that make rather than like phosphorylating proteins, they actually make second messenger molecules. But I think correct me if I'm wrong, but cGAS is the only one of the innate immune receptors that's actually doing that, as opposed to using enzymatic action to like modify other proteins.
Yeah, so it is, well, for the most part, I'm trying to think, depending on how broadly you want to define like innate immune pathways. But the fact that the DNA sensing pathway like creates a small molecule has a lot of really interesting implications for how like the kinetics and breadth and like and spread of the antiviral state within tissues and organs, and like how interferon is induced, because we normally think of these signaling pathways as being activated through protein-protein interactions. And I think that bias is actually what prevented us from discovering cGAS for so long, even though there were many, many labs searching for the sensor. It wasn't until James Chen's lab like tried to isolate like this activity and like looked at it more from the perspective of like, instead of like doing pull downs and identifying the proteins that are interacting with these particular like protein-protein interactors, let's just isolate the activity and then look for it there. So
very cool.
Yeah, it is unique in its structure. Yeah. So given like how these pathways are structured, like the obvious prediction is that, you know, RNA viruses shouldn't be detected by cGAS. So STING, which is activated upon cGAS producing cGAMP, shouldn't be a factor in RNA virus replication. But the spoiler of like my whole paper was like, spoiler, STING is actually necessary for RNA virus control, and it seems to do it in an interferon independent way. So there are like two like main points there, which is, one, these pathways are not structured the way that we think they are. They aren't just parallel pathways. So DNA and RNA recognition are not just parallel pathways. There does seem to be some sort of integration of, or some sort of input, you know, going across from the RNA sensing pathway into the DNA sensing pathway. And also that interferon is not the be-all, end-all of viral control. And I'm not really sure which of those points people have a bigger issue with. I think it seems to be the fact that STING is activated at all during RNA virus infection, but I do think that it's because they have this implicit understanding that you have a PAMP, you have a PRR, detect it, and you activate interferon. And so like when you say actually control is coming through, you know, a different mechanism, they're like, nope, I have a PAMP, I have a PRR, and I have interferon induced.
Well, and so maybe given that, we should just say like what that dogma about interferon is. So interferon is a secreted signaling molecule. It's kind of like a cytokine, which we talk about a lot on this podcast. But basically, it's responsible. But most cells have interferon receptors, and when they detect interferon, they go broadly into an antiviral state. So they make some changes that make them more resistant to. So they
upregulate a few hundred genes that have a variety of antimicrobial effectors. So you upregulate signaling molecules, you upregulate molecules that interfere with viral life cycle in a variety of ways.
You upregulate the receptors too.
You upregulate the receptors, you upregulate entry inhibitors on the plasma membrane. There's all sorts of things. Like 100 overlapping and like very important proteins like get upregulated, and they seem to inhibit viral replication in one way or another.
But see, as a person who was never studying antiviral mechanisms, I always had a hard time wrapping my head around how interferon was acting to affect replication in that primary cell where it's first triggered. Do you know what I mean? Which is why I think
yes.
I liked your paper so much, because I said, well, obviously there would be some other way to control that primary infection, and the interferons, sort of going out into the world and telling the bystander cells, okay, watch out, we've got an infection here. But, the replication cycle happens so quickly that it just seemed to me to be sort of a funny thing to expect that interferon was the end all be all.
Yes, and that actually is like the one of the intellectual starting points as well for me with this project, was really trying to understand, like, when the first virus enters the first cell, how do you stop replication? Because I think that's a really critical decision point on of like, will an infection be productive, or will it, like, the infection of, like, let's think of this as like an in vivo context, will this infection be productive, or will it burn out? Because
on the other hand, you could make you could make the case, and I think people have made the case, that like any individual cell is sort of expendable in a multicellular organism, and so it doesn't necessarily seem critical that the first cell infected survive, so long as it sends the signal to its neighbors to prevent further infection. I mean, I agree that if you could stop the virus in its tracks in the first cell, that would be ideal, but doesn't seem critical necessarily.
So I would push it. So I agree, like from a health standpoint, or like a disease state standpoint, you can lose the first cell, sure. But with that first replication, you've now increased the viral load by a log or more, a few logs or more, depending on the burst size of like the number of virus particles that are made. So this is like really looking at the initiation and like establishment of infection, and like what do you need to do to stop the establishment. So the first cell possibly is not important. It's those first few cells that are important. But I would argue, though, like in the case of HIV, if the first cell's infected, like you're done. Like that's done. Mm-hmm. Like when you're thinking about, because I think like implicit in what you're saying, Kevin, is like the immune system has enough time. Like you can let the first few cells become infected. You're only going to lose a few cells, but by the time that you have enough time to make a response and then dampen the infection. But something like HIV, that's going to like incorporate itself into the genome and hit like whatever cell type. It's really good at evading the immune system, and so that small period of time is actually very critical. But yeah, anyway.
Intellectual discussions about what should be.
Yeah,
I've missed this.
I will also say, like a caveat before we get started on this paper. So all these infections are done in MEFs. So we're talking about fibroblast cells, so like support cells. I don't extend this observation to immune cells or other cell types. Mostly because like this was enough work.
God, yeah.
But
it's a ton of work.
I mean, we do know that like other cell types can wire these pathways in different ways, and so this might not be true for all cell types, but we do use this as like this. I mean, this is why we look at a model system, and obviously this is done in cell culture. This isn't like an in vivo model, so like. When I say like interferon is only important for upregulating ISGs, interferon stimulated genes, obviously in vivo it actually has many more implications for activating the adaptive immune system, activating, inducing like Th1 responses, making sure interferon gamma is up, getting your NK cells like activated, blah 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 blah. So there's a lot more to it, but I like the simple model, and I like proteins touching other proteins more so than like for this sort of analysis. We're talking about things that touch other things within one cell. So going to an in vivo context is maybe not the right model for asking those questions, though important.
I thought you were going to say the caveat is that you didn't use CRISPR. You're using siRNAs the entire time.
Yeah, well...
But your knockdown looks really pretty.
The knockdown is great, which is why I didn't try to move it to CRISPR. Here's a secret with CRISPR. One, it is not like functionally better than an shRNA knockdown, and you can fight me on that. Here's the other thing with this. Okay. Like CRISPR creates cuts in the DNA, and I could not get rid of this like background ISG induction. And so I just didn't want to, and I had half of this work done by the time CRISPR came around. And because, you see the results today, but like they were started 5 years ago. And
I remember.
Yeah. I still personally believe,
I wasn't really trying to give you shit for it. I was trying to give you shit for it.
Yeah, that's fine.
I wasn't actually trying to start a conversation about it.
I mean, the CRISPR is great. Oh, wait. This is a complete aside. Beep, 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 beep. Breaking news from like 3 months ago. Did you see that CRISPR paper on BioRxiv and then how it like tanked a bunch of stocks saying that people have responses, like antibody response, humoral responses to Cas9?
No.
Oh my God.
I did see that.
We should do that paper next. Sorry. Random aside, we can get back. Don't cut this. But that was really cool. So basically, I would like to say I, of course, knew that this would happen because it's an issue with viral vectors, that you have an existing immunity. Because you're infected with bacteria all the time. Bacteria have Cas9. Their little bacteria bodies explode, and you start to recognize the internal proteins of the bacteria, not just the external ones. And so, like, some large percentage of the population already has like Cas9 recognition. And
but that only really matters if you're doing if you're trying to do in vivo editing, which seems like a terrible idea to do as the first pass anyway.
It was
right.
Yeah, we can go back to a discussion maybe in a future episode.
Let's have this discussion.
But just like a little teaser for the future.
Mm-hmm.
But apparently it was it's only been on a preprint. It's on a preprint right now, so it hasn't even been fully peer reviewed.
Yeah, but preprints contain a lot of information.
Sorry, no, I don't mean that as a dig at preprints. I mean, it had like an effect on the stocks. And so it was interesting.
That's
in that
amazing.
It held so much weight financially. Something on a preprint. Goodness.
Back to Sting. We should actually say what the name of this paper is. The name of the paper is, can you say it in your sleep, Kate? It's Sting-Dependent Translation
Oh, yeah, sorry. I thought you were asking about the preprint, and I was like, I don't know what it is.
No, your paper. Your paper is Sting-Dependent Translation Inhibition Restricts RNA Virus Replication.
Yes.
And the first author is Kate Franz.
Yes.
Way to go, Kate.
Woo-woo.
And last author is John Kagan.
Yes.
And it's in PNAS.
Yes.
The Proceedings of the National Academy of Scientists, which is no slouch of a journal.
Thank you.
That's my favorite journal.
Aw.
I love PNAS.
I mean, I think it's my favorite journal, too.
Because it published your paper?
Yeah,
well, there
exactly.
you go.
I detected in your earlier description a little bit of annoyance or maybe not a complete... you're not completely happy with the reception this paper has gotten. Is that because you went through some rounds of review at other journals and had some issues? No,
we only submitted to PNAS. So I've given a lot of talks about this project and posters and whatnot. And I went to one meeting where every single person told me I was wrong. Which is really hilarious, if not disheartening. Every single person was like, you are wrong. This does not happen. This is an artifact, which, maybe it is an artifact, but it's, it at least holds up in these cells, and pretty much everyone uses MEFs for innate immunity. So, but
seems pretty robust to me, but.
Pardon?
I said it seems pretty robust to me.
Yeah. Oh, yeah. But I just laughed at, I received a lot of pushback when I originally started working on this. I think because I'll explain, like, this, the original observation has been like really discounted within the field. People many people have observed that STING pathway is necessary to control RNA viruses, but no one knew why. No one really cared to know. And I think when I started talking about it, people were like, it's a thing that we've seen and we don't really care. We've decided as a group to move on.
That's too bad.
Awesome.
I do feel that I'm being, the response to this has not just been haters. I don't want to characterize in that way. There have been a lot of like really great, professors and postdocs and people who have given me like a lot of great feedback and like helped me like refine the idea, design experiments, and who have been really, really supportive. So I don't want to, just give a shout out to my haters. I didn't do this for the haters. I did this because of all the people who supported me. Thank you to the Academy.
So with that, maybe you should talk through figure 1.
Let's discuss the controversial data.
No, it's not controversial. It's very much respected and accepted by everyone in the field.
Well, it's been peer reviewed and published now, so clearly it must be right.
Yeah, exactly. Okay, yeah. So first figure. So this is where we start to define the phenotype that we saw. So we take MEFs, as Kevin mentioned. We transduce them with these shRNAs that will knock down the RNA expression or the mRNA expression, so it'll stop the protein from being expressed. So we have one that doesn't target anything. It's just a non-targeting, small, short hairpin RNA targeting nothing, and then one that targets STING. And we see that we get really good depletion of STING. I didn't make up this shRNA. I got it from someone who'd published on it like very extensively. So I was pretty confident about this guy. And then what we do is, as we expect, when we take these MEFs and we transfect them with DNA, so we stimulate the CGAS STING pathway. In the absence of STING, you don't have any interferon induced. But if you transfect the cells with RNA, so this stimulates the RIG-I pathway, you see that there's no difference in the induction of interferon. So just as we would expect, you have interferon responses only in the presence of both a PAMP and a PRR. So
just because we've used this, and I know you defined it earlier, but I think it's going to come up again. So PAMP, pathogen-associated molecular pattern. This is the ligand from the microbe that triggers an immune response. And the PRR, pattern recognition receptor, is the thing that detects the PAMP and induces the response in the host cell.
So looking at this, I realized that I have not looked at this since I redid this figure in the resubmission.
Uh-oh.
Oh, no. I was like, what did we put in the first figure? Because this went through so many versions. Yeah, so if you look at figure 1D, that's where we start to look at what does virus replication look like in the presence and absence of STING. So in the presence of STING, in our control cells, you have low levels of virus replication. Mostly the virus is controlled, but in the absence of STING, you see multi-log increases in virus replication. And the thing that's really interesting about this, at least to us originally, is that you have no defect in the ability to induce interferon. So that response that we said was absolutely necessary to control virus replication, at least doesn't seem to be sufficient to control virus replication. So the RIG-I MABS pathway is activating interferon just fine. It doesn't seem like STING is necessary to induce that interferon like we would expect, but the viruses are still not controlled. And then in the figure 1F and 1G, it's the same conclusion, but it's looking at different viruses using Western blots. And again, we don't really see any difference. We don't see any dependence on STING for inducing interferon. You have very good induction of interferon when the virus is one that induces interferon. So this is like the big takeaway with this figure is, one, STING is needed to control RNA viruses, and it's needed to control diverse RNA viruses. So it wasn't just like one type or one strategy of replication that STING was effective against. It's effective against VSV, which is a negative stranded RNA virus. It has a very different replication strategy to something like SynBis, which is a positive strand. It's necessary for Sendai, for flu, for reovirus. These viruses differ in the location that they replicate, and the location of the cell that they replicate. They're different in their ability to induce interferon. So we use a mutant of VSV that induces interferon quite potently due to a mRNA export defect. So like we can't. When I originally did this, I was like, let me find like the type of virus that this is effective against. I'll find the one that Sting isn't necessary to control, and then I'll be able to compare and contrast and figure out the difference, and then have the mechanism. And this will be done in a year. But basically, every virus we threw into this always replicated more efficiently in the absence, or replicated to a higher extent in the absence of Sting.
Can I ask a question here? So in figure 1G, you're showing the interferon response, and you actually have a higher interferon response in the Sting knockout. So not only is it the Sting knockout is preventing control, it's preventing control in the presence of more interferon. And so I'm guessing that the more interferon there is just the hypothesis is the result of more viral replication. So you have more cells infected that are producing the interferon.
Mm-hmm. Yeah, so there's more cells infected, there's more virus present, so there's more PAMP present to activate RIG-I and MAVs. Yep.
Right. Okay.
And that's done in the supplement to just show that it's all MAVs dependent.
Got it. Okay. Awesome.
Okay. So second figure. If anyone wants to jump in and describe a figure, I feel like this is going to turn into like a seminar, because I've spoken about this so much.
Well, that's not necessarily a bad thing.
Okay.
You're doing great.
Thank you.
She's got practice.
So in figure 2, we looked at the biochemical markers of activation of the CGAS Sting pathway. So the hypothesis here is, from figure 1, we know that Sting isn't necessary to induce interferon, and you can consider interferon induction to be the terminal action of these pathways. But perhaps there's a non-canonical activation upstream of interferon induction in the CGAS Sting pathway, and that's how this control is being mediated. So maybe CGAS Sting, they're not activated to induce interferon, but they're activated to do something else. So we looked at all of these biochemical markers of CGAS Sting activation, if that makes sense.
Mm-hmm.
Okay, so the first figure, we look for the most upstream activation of CGAS Sting pathway, so the production of CGAMP, that dinucleotide we talked about. We don't see it made during viral infection, during RNA virus infection, which is what you would predict. We also looked at the movement of Sting inside of a cell. So when Sting binds dinucleotides, it will move from the ER to these perinuclear regions around in the cell. And we see that this happens when you activate with DNA. But if you activate with RNA, or you infect with an RNA virus, Sting doesn't seem to move. At least it doesn't seem to relocalize in the same way. We can't really say it doesn't move. I didn't test for all of that, but it doesn't go to the same place it goes to when DNA is present. And when Sting leaves the ER, it's phosphorylated and eventually degraded, which in this Western blot, we see this phosphorylation of Sting, the degradation of Sting in the presence of DNA. But if you add RNA to the cell, or you infect with RNA viruses, Sting doesn't phosphorylate and it's not degraded. It's actually upregulated because Sting is itself induced by interferon. So it's one of those interferon stimulated genes. So we see this increase of Sting.
I was going to ask about the increase.
Yeah.
Okay.
Yeah.
That makes sense. Yep. And the phosphorylation, that's that sort of that faint band we're seeing at 1 hour. Is that right?
Yes.
Above the main band. Okay. Got it. Cool. Very nice.
Yeah. Western blots were not my favorite thing to do in grad school. So
I didn't
Can't imagine why.
I didn't actually ever do a Western blot until I got to my postdoc, if you can believe that. I got through all 5 years of grad school without doing a single Western blot. I did a crap ton of Northern blots and primary extensions, but no Westerns.
I think we've told this story before on the podcast, but my favorite Western blot story is from Matt, who rotated briefly in the Kagan lab, and like literally caused the transfer apparatus that we use for Western Blast to start smoking.
Oh my God.
Because he did like 5 volts instead of 5 millivolts or something like that.
Oh no.
And John walked in as the transfer apparatus is sort of smoking and just like walked in, stared at him, and turned around and walked out. And Matt to this day has not done another Western block. So
that'll do it.
And that's why he didn't join the lab.
But so Matt and I have talked about this. We probably talked about this on the podcast as well, but
I'm sure we have.
When I was rotating in the lab, that same transfer machine, I broke. I dropped it. It just slid out of my hands and just shattered on the floor.
Oh my God.
The little screws that hold the electrode in place or whatever it is, just sheared right off. No saving it. And I had been told, like, there was this grad student a few years ago. He wasn't allowed to join because he really messed up this the transfer apparatus, and I just hit it.
Wow. It sounds like that transfer apparatus is cursed.
Or that transfer apparatus gives a really hard time to the best scientists among us.
Mm-hmm.
Matt and
That's
I.
a beautiful way of looking at it. I like that.
And it gives a harder time the better scientist you are.
Mm-hmm.
So
I broke it.
Straight up shattered when it took one look at you.
Yeah.
I should say, I don't think that John would have prevented Matt from joining the lab due to the transfer apparatus. I think that Matt was just so embarrassed by the whole thing that he decided he couldn't show his face again.
Oh, yeah. No, I think
And Kate has no shame, so that is not a bar for her.
Well, I hid the apparatus, so I did have some shame.
I see.
Oh no!
I mean, now that I know the lab, I don't think it would have prevented me from joining. But at that time, I was on like my 5th rotation or my 4th rotation, and I just really wanted to join a lab, and I really liked the lab I was in. I was like, I can't let anything take me down. I gotta hide this. So yeah.
Secret's out now.
All right. Well, John's not going to listen to this podcast, so
never know.
I think I tried to get him to listen once very early on, and he was just like, "No, it's not going to happen."
Good.
I don't want to deal with that. I can just imagine John listening and screaming into his headphones. What is wrong with you?
Or just getting like an email of like, this is how you should explain this topic. You're not transitioning right. Anyway, now we're going inside baseball. Okay.
Yep.
So let's see here. Where were we? Okay, so
Figure 3.
CGAS and Sting, they are not active during RNA virus infection. So they're not activated canonically in any way that we have characterized for DNA viruses. So now that I talked about transitions, I somewhat forgot the transition into this part.
Well, so here I think now at this point we're left with 2 possible explanations for why we're seeing higher viral replication in the absence of Sting, right? One, that the rate of viral replication is higher, like Kevin was saying. You've got more PAMPs to be recognized, or 2, that establishment of the infection is more efficient in the absence of Sting.
Yes, that's exactly right. Okay. So we tried to deconvolute. So the thing is when we infect in the presence or absence of Sting, we're looking at viral gene expression. So we don't really know what the infection looks like on a single cell level, and so we can't really make any claims to, is it that the virus is going from one cell to the next cell faster in the absence of Sting, or is it starting at a higher level of infection? Yeah, you explained that beautifully. I don't know why I'm re-explaining it, but so we infected the cells. We did a lot of viral infections, and then we did this linear regression analysis to determine what is the growth rate. So these are just your normal exponential growth rate equations, and you can use them to model the virus replication. So any exponential process, you can use these equations to model. And then what we do is we fit the data to the equation, and then we extract what the growth rate would be. So that's just like, basically it's a constant will be in that equation. And so when we compare what the growth rate is predicted to be in both of these infections, we see that the growth rate is not statistically different. So the growth rate doesn't seem to be different. So the growth rate seems to be possibly the same, but it's not different, which makes sense because when you look at so we had to do these infections at higher MOI, so you could actually see what the growth rate is in the presence of sting. And when you have productive infection, you can see that these lines are basically parallel to each other.
Mm-hmm. What MOI did you use? Just curious.
1.
Oh, okay. Yep, I see that.
Yeah, so the earlier infections were done at 0.05 or 0.1.
Got it.
So then we said, okay, well, perhaps at the start of infection, there are more cells infected. So when we do these infections in the lab, we take monolayers of cells and we put a virus inoculum on top of it. So there are viruses that we've grown and we've isolated and purified. And you put the viruses on top of the cells, and then you let them basically enter the cell and start replication. And you take all the extra viruses off and you throw that away. And then you let the infection proceed. So in that first initial step of infection, we thought perhaps there are more viruses getting into cells. And that's why we see this higher level of viral gene expression. And so the way to measure that is by using a plaque assay. So you can look at how many virus events, like how many viruses get into a cell and then induce and start productively replicating. I will say that this is not a measure of viruses entering cells. That is a different and more difficult analysis to make. This is just productive infection, plaque forming units. And we're using this as sort of a stand-in for entering a cell and establishing infection. Obviously, like you could maybe enter the cell, you establish infection in one cell, but then it doesn't go out from that cell. You would never see that plaque. But actually, in this case, you would, because I counted these by eye on using JFP. So if you infected one cell and it was productive, I did see it. So if it didn't grow into like a giant plaque, I still could count it. But if if the virus got in, it started replicating and then stopped for whatever reason, I had no way of knowing that happened. So that is the caveat with this. But it still tells us that at least producing these productive infections that go on to create plaques occurs more readily in the absence of sting. So we say that sting decreases the infection probability. So if sting is present, then then the virus is less likely to productively infect that cell.
Got it.
Yes.
Well, and you sort of get at the entry question a little bit in panel D, because you sort of bypass the natural entry by using the lipofectamine, right?
Yes, absolutely.
So that sort of rules that out.
Yeah. So that's one of the ideas here is, well, if the cell is more likely to become infected, perhaps it's because entry is more efficient or occurs at a higher rate in the absence of sting. So to get around that entry, so sting could somehow inhibit entry, and that's how this entire mechanism works. So the way that we got around that was we took viruses, VSV, we isolated the minimal infectious unit. So that's the core of VSV, if you know anything about VSV. And then we can transfect that into cells and then see if the viral gene expression after a few hours is different or the same. So if it's the same, that means that entry is likely a factor because you put in the same amount of virus, you bypass the normal entry pathway, they make the same amount of viral gene expression, so the block seems to be at somewhere around that entry point. And then that would require, the really fine, highly sensitive experiments that I did want to avoid. So I was not sad when I saw that the change in gene expression actually wasn't affected by entry. So you transfect in the viral cores and you actually get more replication, more gene expression in the absence of sting. So sting isn't only, we could say that the main function of sting is not to inhibit entry.
Got it.
Oh cool. Boy. Now we just get into like these Mondo figures. So we can just start skipping things.
Yeah, these are huge.
Right. So maybe worth summarizing at this point.
Yeah.
So we have this protein sting, which is known to block, or is known to basically induce, maybe I'll call them like standard antiviral responses to DNA viruses, like producing interferon and doing all the things we normally associate with PRRs. You found that despite the fact that Sting is not doing that for RNA viruses, you nevertheless have a block of viral replication, RNA viral replication from Sting. And you've now narrowed it down so you know that it's not affecting the growth rate of viruses, but it isn't affecting the ability of viruses to establish infection in a cell.
Yes.
But not because the viruses can't enter, or not solely because the viruses can't enter cells.
Yes.
So maybe rather than stepping through all of these other figures directly, maybe like at this point, when you have this information, what are the hypotheses about ways that Sting could be affecting a virus infection that isn't slowing the growth rate or blocking entry?
Yeah. So,
or like what's the next, or producing interferons or what have you. So what are the possibilities left open at this point?
Yeah. So what we hypothesized was that Sting must affect some viral replication step that is common to all viruses. So we know that viruses will still enter the cells in presence or absence of Sting. There's something after that entry step that Sting is doing to stop viral replication, stop viral growth. And so one thing that's shared among all viruses is that they're all dependent on host translation machinery to translate their mRNAs. So the viruses cannot replicate, they cannot make their proteins, cannot undergo their life cycle events if they do not have access to the translation machinery. So that's your ribosomes and amino acids and all that stuff. So we saw in the literature that there had been some work done that showed that the more efficiently a viral mRNA could engage with a ribosome, that correlated to higher infectivity. And so that gave us the idea of looking at translation of RNAs in the absence and presence of Sting. So is that different? And if you have Sting in the cell, do you still engage the ribosome in the same way? And so what we did was we transfected cells with reporters. So basically we made RNA outside of the cell, and it coded for luciferase, and then we transfect it inside a cell either with or without Sting, and we looked at how much luciferase was made. And what we saw was, like, in a normal mRNA, you have this increase in luciferase expression in the absence of Sting. So it says that Sting is necessary to I mean, you could say it in a couple different ways, but in the absence of Sting, you have this enhancement of translation. So you have more protein made, even though you have the exact same number of RNAs in the cell. And the RNAs had the same stability, so we were pretty confident that it was a translation issue. And so we tried to identify exactly what in translation was different in the presence or absence of Sting during these RNA transfections. And we couldn't do that so well, except we have some data where we look at different, so we transfect with RNAs that have different, boy, what's the way to describe this? I haven't thought about this in a while. So you know what? I don't want to talk about this.
I love your VSV construct. I thought that was a really neat virus.
Yes. Oh, I guess I do have to talk about this if we're going to talk about. Okay. So
yeah, no, this is cool. So you have different RNAs that you can transfect in, one of which encodes for firefly luciferase, and one of which encodes for renylluciferase.
Yes.
Right. So these are enzymes that will make a chemical reaction that produces a luminescent glow in the presence of, I forget, how are they different? Is it just the wavelength of light that's produced is different?
Something
wavelength is different.
Not produced. It's the one that excites it is different.
No, I think it is. Well, so
it's not fluorescence.
I'm thinking fluorescence.
It's
I'm sorry.
So Firefly is so first of all, there's size difference. So Firefly is 61 kilodaltons, and that will produce a greenish yellow light in the 550 to 570 nanometer range. And then Ranilla is smaller. It's 36 kilodaltons, and that's blue light at 480.
Okay.
So they are different. That's why they're so useful in the same system.
Right.
And so that's what's so cool about the virus is that it's actually encoding both at the same time, but it's the control of translation that's different because Firefly is driven by traditional cap-dependent mechanisms, but the other one is this unusual IRES, right? This cricket paralysis virus. Is that right? Or is it the polio, actually?
No, it's the cricket. Yeah.
Okay.
So this was But
regardless, the iris is internal ribosomal entry site, so that basically allows the ribosome to engage the RNA without using the typical 5' cap driven.
Yeah.
Right.
Is that right?
Yep.
Yes.
Yeah. So okay. So what I was hesitant to explain, but now you got me halfway there. Is that, so when we use a typical cap, so this is what initiates the ribosome selection, loading onto the RNA. We see that there's more protein made in the absence of sting. And now viruses, they don't always have these structures on the ends of their mRNA. Some of them do. There's a lot of variety in how mRNAs are translated by viruses. But one mechanism is through these highly structured RNA stem loops, and it's called an internal ribosome entry site. And this can engage the ribosomes, and they use limiting numbers of translation initiation factors. So you have the ribosome, but you also have 40 different proteins that are involved in positioning it and getting it ready to go and making sure the enzyme part of it is working and blah, 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 blah. It's this very coordinated, complicated, beautiful process. And viruses have found ways to cheat and get away with not using every single one of these proteins. And so you have something like poliovirus, which I used a poliovirus iris to drive luciferase. That one will use pretty much everything, every single protein, but one protein. So it's very similar to the way mammals and humans will translate their mRNAs. It just can get around without one extra protein. And then on the other end of this is the cricket paralysis virus. And cricket paralysis virus doesn't use any translation initiation factors. It can grab a ribosome directly and start translation. And so what we wanted to see was, since there's these ranges of irises that use decreasing amounts of these factors, is there one iris that isn't regulated by sting? And that would tell us what factor sting might be regulating. But what we found was the only iris that wasn't regulated by sting was this cricket paralysis virus, which basically said sting probably regulates a translation initiation factor because cricket paralysis virus is not regulated by sting and doesn't use any translation initiation factors, which is interesting in itself, but allowed us to utilize this really cool virus, which basically what we were able to do is make a virus that let us read how much viral translation was occurring on each mRNA. Okay, I feel like I will have lost people at this point.
I think that's good. I think I would recommend to people trying to follow along. They made a beautiful construct in their paper on Figure 4C. If you want to have a look at what this looks like, it's very nice.
So essentially, what we did was we put in this bicistronic mRNA. So bicistronic means it has two coding regions. So the first part of it is going to have the normal VSV 5' end. It's a cap that initiates translation, and that will make Firefly luciferase. And then the second part is CRPV iris that drives Ranilla luciferase. And so because CRPV iris is not regulated by sting, all of the Ranilla signal we get in this virus is going to be equal to the number of, is going to be related to the number of mRNA molecules in the cell. And all of the signal from the Firefly is going to be the amount of viral protein produced. So we can make a ratio of viral protein produced, that's a firefly signal, to number of mRNA molecules in the cell, and that's the vanilla signal. So we've made we've constructed this efficiency of translation measurement.
That's cool.
And
I like it.
That part was very cool. I did not make this virus. This is Bill Niedermeyer in Sean Whelan's lab, and he's wonderful. He also taught me all of my translation stuff. I love him very much. I called him my lieutenant many times. I think he asked me to stop. But he is lovely, and if you're looking for a postdoc, you should hire Bill because he's wonderful. So we were able to make all these translation measurements in the cell. And so we found that similar to what we saw when we transfected in mRNAs into a cell, viruses, VSV translation was higher in the absence of sting. So we had more proteins made per mRNA transcript in the absence of sting. And so we measured that to be around 2.5. So we in the first few hours, that ratio is 2.5. So we're like, okay, there's 2.5 proteins made per transcript. It's not really per transcript. It's, just an estimate. But it is a really cool tool. Okay. Oh, and something else that I've done throughout this paper is we also look at knockout cells. So this isn't an artifact due to our shRNA. This is something that is also present if we look at sting knockouts. Okay. So this next figure
was a lot of work.
Oh my goodness. I can't even
Did you do the polysome profiling yourself?
Yes.
Did it eat your whole life?
Yes. It was the worst thing ever. I can't even explain how many hours went into figure 5A, B, C, and F. Oh my goodness. I'll just start crying.
Well, so maybe we should just gloss over that figure
I think we
because
should.
It was so much work. But maybe just broad strokes.
Yeah, broad strokes.
A
So
polysome is
the virus,
where
the VSV CRPV bicistronic virus that we made, it suggested that translation was more efficient or higher in the absence of sting. So we wanted to use real translation assays to show that. So we did this sort of analysis called a polysome analysis. I'm not going to get too deep into it, but essentially what you do is you lyse cells, you separate the ribosomes by size. So you have one ribosome at the top so you put it on a gradient, and a sucrose gradient, and so it separates the ribosomes by size. So single ribosomes are at the top, and then when multiple ribosomes are on one transcript, that's heavier, so that's at the bottom. And essentially what it allows you to do is you can look at where the RNAs are within that gradient. And so the further down an RNA is, the more ribosomes are on it and the more efficiently it's being translated. So our hypothesis is in the absence of sting, there will be more transcripts that are further down this gradient. So there's more transcripts that are being efficiently translated. And that's exactly what we saw. So we saw, in this bulk measurement, in the absence of sting, if you infected them with VSV, there were more VSV transcripts in these heavier polysomes. So we then looked at the amount of translation that was happening on a single cell level using, you just label the amino acid. So it's, so basically we can make an estimate of how much protein is being made and then analyze it by flow cytometry. And if you infect cells with VSV, in the absence of sting, there's more proteins made than in the presence of sting. Okay, so virus translation is more efficient in the absence of sting, and then we try to figure out why and how. So we used one of the things that is pretty common with viruses is that they manipulate the translation machinery because they want to be translated. So the host cell has all of these responses to try to stop viruses from utilizing ribosomes. And so we wanted to look at what does translation look like during RNA virus infection, but we didn't want to have to deconvolute how much of this is due to our RNA viruses manipulating translation and RNA viruses making proteins. So we use a model, poly-IC, that's of viruses entering, and we looked to see what the virus, what translation was doing in the presence of RNA. And again, we see that translation is higher and more efficient in the absence of sting. So when RNA enters a cell, even if that RNA isn't coding for anything, all of the host proteins that are being translated also will be translated more than the host proteins in the presence of sting.
Okay.
So last figure. This is my favorite figure. It took me 2 weeks.
Nice.
Is it your favorite because it took 2 weeks, or?
Yeah.
Okay.
So in this, so I glossed over some of like the nitty gritty translation stuff. So I'll continue to gloss over it in figure 6. But
EIF 2A phosphorylation, blah, 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 blah.
Yeah, all of that stuff. I mean,
great.
It becomes like letter soup, and it's really cool.
Yes.
But the take home message of that is we weren't entirely sure how sting is regulated, like what actual initiation factor sting is regulating. But what we did start this whole, many moons ago when we started recording, what we did start talking about was like this dogma of, control and interferon. It's like control meaning interferon isn't induced unless the PRR can recognize a PAMP. And so even though those are the rules for interferon induction, we thought maybe these are still the rules for whatever sting is doing to control. So this translation inhibition. So we hypothesized that because RNA viruses, their main PAMP is RNA, perhaps RIG-I is what recognizes the RNA, and that RNA RIG-I recognition is still important to initiate the sting-mediated control. So we do this genetically, meaning we don't show that RIG-I and sting interact directly through chemistry, but we do show genetically through knockouts that in the absence of RIG-I, we still have the same translation phenotype. So viral translation is higher in the absence of RIG-I. The host translation is higher in the absence of RIG-I in the presence of RNA. And what we then look at is this analysis of, okay, if RIG-I and sting are in the same pathway, then in the absence of both RIG-I and sting, we should have that should look the same as a RIG-I knockout. If RIG-I and sting are in separate pathways, we'll have one sort of viral control phenotype in the absence of RIG-I, but if we knock out sting, we'll have like even less control. We'll have even more replication. So what we did was we took our RIG-I MDA5 knockout MEFs, we knocked down sting, it's a functional knockdown, and then we infect them with virus, and we see that the replication is the same in the presence and absence of sting. So we can genetically construct this pathway that RIG-I recognizes the RNA, and then it does something. Sting will stop the translation. So all in all, we see that like even though the control mechanism isn't this induction of interferon, the same principle still applies that you need to recognize RNA, and then sting will control the RNA virus replication.
Okay, that makes sense to me.
Cool.
I'm just slowly piecing that together in my head.
I've
never been good at like pathways. That's always been a struggle.
Yeah, this one.
But I think that makes good sense.
This one's a lot. It's really good. The thing about this paper is that it's really like 3 papers, and there's like 3 big transition points in it, and it requires just a lot of expertise in 3 different things.
Yeah,
Right.
no, it was this huge paper for sure, and I can see how much work must have gone into this. So one question I have for you then is, with all that being said, then what do you think sting is doing exactly to repress the translation here? How do you think that's getting engaged and activated?
Yeah.
I mean, that's like the big black box,
It is the big black box. Okay. So, I mean, the short answer is I'm not really sure.
Fair.
I mean, the easiest model is that sting somehow regulates a translation initiation factor. And so when RNA is is there. So like when RNA is detected, so when, viral RNA is detected, Sting somehow is restricting the access of some translation initiation factor, and the translation is stopped.
Has anybody ever done pull downs with Sting to see what it's associated with? Or
yeah,
yeah,
I'm sure.
And so that's difficult because usually when you do a pull down, you filter out all of the translation initiation factors because there's so many of them. You get
Well,
all sorts of factors.
I see.
Yeah. So there are some, if you look at people's math, I personally didn't do it. I did some other mass spec for other reasons, but not on Sting. But other initiation factors have been reported to come down with Sting. I looked at them and I couldn't, from levels or amount of protein, I couldn't find anything that was different. But I have this other hypothesis in the back of my head that I haven't quite nailed explaining, and I can try now.
Give it a shot.
Okay.
Do it.
There was a paper that came out about a year ago, and one of the interesting implications of the paper is that this complex called a stress granule is regulated different if the initiation of the stress granule formation is a chemical or is RNA. So to back up, a stress granule is a way that the cell can stop translation of certain mRNAs. They basically take the RNA, you wrap it up with all of these suppressing proteins, and a ribosome can't translate it. And so I thought that this was really interesting because I also have a phenotype where Sting regulates translation, but only in the context of RNA. So I also did other, which I didn't talk about today, but if you do read the paper, I show that other chemicals that can stop translation are perfectly able to stop translation in the absence of Sting. Sting doesn't affect them, but RNA, which is known to stop translation through this pathway of EIF2 alpha, it does require Sting to actually inhibit translation. So I had this thought that maybe because RIG-I is involved, perhaps the stability of this of stress granules in the presence or absence of Sting is different. And so in the absence of Sting, the mRNAs are able to exit a stress granule more rapidly. But I don't entirely know. So I never tested it. Some people have told me no, that's not how it works. But I do think that is an interesting correlation.
But they also told you that your phenotype was bullshit. So
yeah, but
maybe different
the people told me I don't know. So I think it's interesting. There's a lot of ways to drug this to try to tease out something, but I didn't have much success with those approaches. But yeah, So I have thought that maybe Sting regulates stress granules in some way.
You did enough in this paper.
Oh my God.
Yes.
Could you imagine if I did more? There's also a very there's a lot of giant supplemental figures where I talk about all the things that Sting does not regulate that are not translation.
Uh-huh.
But yeah.
Can I ask a slightly different question that maybe gets to the same point, which is in terms of the structure of Sting, so we know it's a DNA binding protein.
No, it's a dinucleotide.
Sorry, dinucleotide binding protein. C-gas is the DNA binding protein, right?
Dinucleotides are actually small RNAs, so it's actually an RNA binding protein. Sting is.
Okay. That's interesting.
Isn't that cool?
In terms of the other parts of Sting, it's ER associated. Is it embedded in the ER membrane, or is it just peripheral?
No, it's embedded. So it has 4 transmembrane domains. Okay.
And is there any evidence from the structure about what other functions it might have? Is there a lot of other structure in the cytosol that could potentially be interacting with stress granules or initiation factors or whatever?
Yeah. Okay. So so the structure of sting from like N terminus to C, you have these 4 transmembrane domains that are embedded in the ER membrane. And you have this linker domain coming out of the membrane. And you have this giant domain, which makes up the majority of the sting protein, which is the cyclic dinucleotide binding domain. Mm-hmm. And then mammalian sting has a C-terminal tail. So it has this little tail. And mm-hmm. So we know what the cyclic dinucleotide binding domain does. It's conserved back to like these early ancestral sting alleles. But the C-terminal tail is really interesting. It's only found in animals, or it's only found in animals. It's only found where there's an interferon system. So this tail co-evolved with the domain of cGAS that allows it to detect DNA specifically. And this tail is responsible for coordinating a transcriptional response. And so we had, so I didn't publish this. So like, that's a thing to like remember is, so we did some experiments looking at the ability of this tail to either regulate translation or control viruses independently of interferon. And one of the things that I think is really interesting about this tail is it seems like to fold in on itself when it is inducing interferon, so it gains some structure. So I kind of think of it like an elbow. So like you have like your arm extended, your hand is like reaching out, and then when interferon is inducing, your hand comes towards your chest, and then the transcription factors that induce interferon kind of sit on your elbow. But if interferon is not being induced, this tail is thought to be unstructured and is like free flowing. So I think that there's a lot of research that can be done into like, are there multiple functions of the tail of sting? It's kind of like a cool little guy. And also, I don't know if I should say this, but I'll say it to you guys. I'll say it to you guys. Also, so one of the things that people say is like, oh, the tail is only present in animals, because like, insects don't have interferon. Interferon evolved later. But in flies, we can talk about flies. There's a whole part of this project that was done in flies that I did not publish on. In flies, the fly sting actually does have a tiny, tiny tail. And I wonder if that's, and it doesn't have interferon. So in that tail, it's much smaller than the normal sting tail. But I do wonder if that tail is necessary to control viral infections in the fly in a sting-dependent way. And that would be cool. That's an interesting thing to look into. So that's why I'm interested in the tail in particular. But that would be interesting. Also interesting.
Well,
one more thing.
Oh, go ahead.
One more thing about the fly. Fly sting is not thought to bind dinucleotides, but is thought to be functional in antiviral responses.
But it's got the giant dinucleotide binding domain or something homologous to it?
Yeah. So perhaps if, this function were conserved into flies, you could start trying to figure out what about it. It'd be an easier way. It might be easier to figure out what part of sting is actually necessary because you don't have to deal with the effects of like interferon.
Right. Cool. Well, if anyone, current grad student or postdoc, is interested in pursuing this and trying to scoop whichever of John's grad students comes next and picks up this grad.
It's continuing on my absence.
Have you already dug a big enough hole for yourself at the beginning of the episode? Just throw it all in.
Like I said, John is never going to listen to this podcast. There will be no evidence. If you have any interesting ideas, you should let us know. You can get in touch with Kate and give her your crazy ideas about Sting's tail at comments@audiommunity.org. You can find the podcast as well as links to all kinds of crap, including the things that we're drinking. Kate, you need to send me a website or something for the whatever abomination you were drinking
the
earlier.
delicious rat.
And you can find all of that at emmunity.org, emmunity.org. We're on Facebook, facebook.com/Audiommunity. If you would like to support us making this show, you can toss us a few cents, up to $1 or $2 per episode, by going to patreon.com/Audiommunity. There you can set a donation amount, the maximum amount you want to give in a given month. We've only been averaging like 1 or 2 episodes per month at this point. Hopefully we can increase that, but you can set maximums, and then we get a little bit every time we release an episode, and it's really great. We're trying to come up still with rewards for our patrons. Kate is going to critique your poster. If you want her to, you're going to get a shout out when you buy a beer that we drink on the episode. Anyway, we're going to figure that stuff out. If you have ideas, you can send those to comments@emmunity.org. If you've got paper suggestions, you can send those to us as well. What else? We're on Twitter. Kate, I still haven't. You need to add your Twitter handle to your bio on the website.
Oh, okay.
Chadene is also on Twitter.
I am
at
Chadene Tremags. That's
easy.
It's on the website.
I'm Kev Bonham. I think Camilla's on there too. But anyway, you can find everything at emmunity.org. Also, you can support us on Patreon, but the best way to support us is to share us with your friends. Tell the other grad students, postdocs in your lab, even technicians, that they can learn a little immunology, have some fun by listening while they're doing tissue culture or trying to get polysome profiling working, or setting transfer apparatuses on fire. Any of those are perfectly legitimate times to be listening to Audiommunity. So share us with anyone that you think would be interested. I think that's it. With that, we'll see you next time.
See you around.
See you later.
Sorry.